Hello, everyone. My name is Dean Butterworth. I'm the Outreach and Education Specialist at Olympic National Park. Welcome to the final perspectives in our series this year. Thank you for coming out on a beautiful day. We're very excited to have our speaker with us today. A few things that are going on at the park that we'd like to add to, in addition to some of the announcements from the library. The week of April 20th is National Park Week. So April 20th is a fee-free day at the National Park. So you can come and visit the National Park and there will be no entrance fee charged on April 20th. It's also the the Washington coastwide cleanup on April 20th. And so you could go to our website. You'll find links there on how to register if you want to help us to pick up marine debris on the coast. If you don't want to go all the way out to the Pacific coast, there are sites along the Strait of Juan de Fuca that you could sign up to help clean as well. Saturday, April 27th is Junior Ranger Day at the Olympic National Park Visitor Center. We're super excited about Junior Ranger Day this year. There looks like there's maybe one junior ranger in the audience, but you might know other junior rangers. So we would like to have you invite those junior rangers, bring them along with you to the visitor center. We're going to have a climbing wall. We're going to have the mules that help to keep the trails open. The fire engine will be there, a ranger patrol rig will be there, the Friends of Olympic National Park who've providing us with the refreshments today. Thank you, friends, for the refreshments. They will be there as well. Faro Marine Life Center, Olympic Coast National Marine Sanctuary. It's going to be a great, big, huge event, and we'd like for you all to come and bring your junior rangers with you for that. And so that's the events that are coming up in April that we're really excited for. And we're also really glad today to be offering this presentation. The library is such a great partner. They make it possible by providing the room and providing all of the technology and recording the presentations so that if you miss them, you can go back to the website and you can see the previous recording. So we're grateful to the library as well as to the Friends and our other organization that helps us with the perspectives presentations is Discover Your Northwest. And so we're grateful to them as well for all of their help to support this program. And tonight we're really fortunate to be able to hear from Rebecca McCaffery. She's a research ecologist with the U.S. Geological Survey, and I'll let her introduce herself and just say thank you for your time, for coming here, and for sharing your knowledge with us.
Okay, thank you. And everybody can hear me okay in the audience? Okay, wonderful. Yeah, so my name is Rebecca McCaffery, and I run the USGS Olympic Field Station here in Port Angeles. And this is part of a larger USGS Ecosystem Science Center called the Forest and Rangeland Ecosystem Science Center, which is headquartered in Corvallis. So I'm going to talk to you today about bats on Washington's Olympic Peninsula and a little bit across Western Washington as well. And of course, like all scientific endeavors, the work that I'm going to share to you today is really a collaboration between numerous people and different groups. And so I have listed some of my primary collaborators here on the opening slide, and just want to acknowledge that this is part of a much larger group of people. So we have folks at the USGS working with me, and then the Park Service, of course, and Tara Chestnut in particular, who was the wildlife biologist at Mount Rainier for several years, kind of spearheaded some of the initiatives and some of the work that I'll talk about today that's occurred across all of our parks in Washington, as well as Oregon State University Cascades, which is the headquarters of the Northwestern Bat Hub and is one of our partners in some of our regional work as well. So to orient you today, what I'm going to try to do is I'll just start out and kind of get us into the world of bats, talk a little bit about what bats are all about and their role in the ecosystem and some of the threats they're facing and how we study them. And then I'm going to get into some details about some of the things we've been doing over the past several years here, especially focused in Olympic National Park. Okay, so bats are really cool. They exist on every single continent except for Antarctica, and they comprise 1/5 of all the mammalian species. So they're really more diverse than the most diverse order after the rodents. They also are really the only mammals that can fly, which is super cool as well. And they're masters of echolocation, which is the way that they find their prey and orient at night. They're really ecologically diverse as well. So we have bats that here in our Pacific Northwest systems are insectivores. They're eating insects. But around the world, bats eat pretty much everything. We've got fruit bats. We have bats that are pollinators. We have bats, the vampire bats are sanguinivores. And we have carnivorous bats that eat everything from frogs to fish to birds, and even bats that eat other bats. So really diverse, you know, and you can see here in this kind of examples of their different ears in particular, which are adorable. They're also extremely long-lived. So bats, you know, there's so many bats are so tiny, and most of mammals that you think of that size, your mice, your shrews, you know, they're just kicking around for a couple of years, but bats can hang out for a long time. And the example I have here is this Brand's myotis that weighs less than half of a pet store mouse and yet lived for over 41 years. It's like 10 times longer than what you would expect for a mammal of that size. And they're a really important part of healthy ecosystems. I mean, all species are precious, and bats are part of really dynamic food webs. When they die or if they decline, you know, other parts of the food web are affected. So it can be everything from even cave-dwelling bats. There are organisms that rely on their guano for nutrients, and that's their bat droppings. Fruit-eating bats in Pacific Islands and Latin America will disperse seeds, and so they play a really important role in the plant dispersal. And then several animals, including hawks and falcons and owls, weasels, ringtail cats, rely on bats as their prey. And so bats provide sustenance to other species as well. I pulled out a couple kind of examples of what we call ecosystem services. So things we can almost describe an economic value to that bats bring to our lives. They eat a lot of insects, and it's been calculated that they actually provide $3.7 billion worth of insect control per year for US farmers. So that's just in this country. So if you have a loss of those species, that really changes an aspect of how agriculture works in this country. And then I also mentioned the species that are our pollinators. So one fun example is the species in the Southwest that pollinate the blue agave, which is very important to tequila. It's primarily done by bats. But, you know, bats also are very vulnerable to a variety of threats. One of the things about bats that make them super cool are also things that make them vulnerable. By being really long-lived, the trade-off of that for a lot of species is that you don't have as many babies. And so that low reproductive rate, that they're not having a lot of pups every year, like you think of mice, they're just reproducing all the time, and rabbits and things like that. That means that their populations don't change at the same rate. And if you have adults dying, then, you know, there's less pups coming along. They also have a really high metabolic rate. They need to eat a lot to fly. And they face a lot of threats that are human caused. And some of these we hear about a lot, things like habitat modification, climate change, human persecution, because bats, for some reason, I don't know why, are scary to people. But there's also some bat-specific threats. that have really kind of come on board. And one of these is wind farm collisions. So there have been some really high mortality rates from collisions with wind energy turbines. And another one is white nose syndrome. And this is an infectious disease that I'll talk about a little more in a couple slides. Another thing about bats is that they're relatively understudied. And I'm going to talk a little bit about this in the context of the ways that we've tried to learn more about their ecology throughout the talk. They're nocturnal. So, usually we're kind of asleep when they're active, although some humans are pretty nocturnal. They're small. A lot of bat species are really small, especially the species around here. They're cryptic. They're often just really hard to find. They can tuck into the tiniest little crevices in your roof or in just under a little tiny piece of bark or a little rock on the ground. And they can move around a lot. And so you're also trying to deal with an animal that could move, miles each night when you're trying to study it. So all this can kind of be a bit of a struggle in understanding these species, and especially some of the species here in the West. So I'm going to talk a little bit more about white nose syndrome because that's been the motivator for some of the work that I'll be discussing tonight. As I mentioned, it's an emerging fungal disease. It's caused by Pseudogymnoascus destructans. From now on, I'll call it PD. And this is a disease that has killed millions of bats in the United States. And it is a cold-adapted fungus, so it can only infect the tissues of bats during the winter when bats go into torpor, which is kind of their state of inactivity. And that's when the fungus can grow. So for about 10 years, white nose syndrome was really restricted to the eastern part of the country, and it has spread now to the west. So I'll give you a couple slides to show that change. So this is where things stood in 2015. And sorry, I'm having like mic things where if I move my head, then I feel like I'm going to talk this way. Oops, sorry. Now I've jumped. You can see the X here. And maybe that's not working, the pointer. The X here is where it originated in upstate New York. And so you can see that spread that's happened since then from 2006 when it was first detected to 2015. And then this huge leap happened in 2016, and it was just discovered for the first time kind of near Mount Rainier National Park. And then now scooching all over to the present, we've actually backfilled a lot of that space between the West and the East and several provinces in Canada as well. So it is continuing to spread and becoming fairly ubiquitous in the country. I'm going to zoom into where we are now. So you can see the spread. So the way that map that I showed you before summarizes the data by counties. So that's kind of zoomed in on the bottom here. So you can see that it has reached the peninsula, although it has not reached the park yet. And it is just slowly spreading out from that original location. This is how, on the upper graph is how the upper map is how the Washington Department of Fish and Wildlife summarizes their data. So you can see a little bit finer where we've detected bats that have the disease. So this has led, to a lot of work by different agencies across the country. The USGS has a National Wildlife Health Center and does a lot of disease work. So they were involved in identifying the causal fungus, PD. They've also been really involved in kind of helping to track and map this fungal spread across the North America and in characterizing the effect of white nose syndrome on populations of bats. And this has been done in collaboration with other agencies like the U.S. Fish and Wildlife Service, the Park Service, state agencies, and of course, academic researchers. So it really is taking a lot of different people to try to come to terms with what's going on here. There's also work responding to white nose syndrome right now. So work on things such as potential vaccinations, probiotics, other ways that we might be able to tackle this disease. And some of the work that we do is just really understanding what's happening with the bats as it relates to white nose syndrome spread. And I bolded that because that's kind of where we're at here in Washington State. In the Park Service, there's been a white nose syndrome response strategy as well. And so in the Pacific West region, which is our region, there's a number of pieces identified to try to educate and provide more data and information and track the disease. And so this is included in training and resources and these communication pieces, but also the bat monitoring and white nose syndrome surveillance. So that's, again, what we'll be kind of moving into focusing on tonight. So I'm going to switch gears now to think specifically about our national parks in Western Washington and how this relates to the emergence of this disease. National parks can really serve as a beacon and a beautiful place for the habitats that are less impacted by by some of the stressors that I outlined in terms of like habitat destruction and other human-caused stressors. These are protected areas. Although they're not immune to things like disease and climate change, they are places where we can kind of look at baseline examples of healthy populations and look at how they are using these forested habitats, in our case, in our Pacific Northwest forests. Now, bats in Washington's national parks were inventoried back in the late 1990s and early 2000s. But these were really kind of standalone efforts. Each of the three large parks did have an effort to kind of see what bats were there and kind of where they were, but it didn't turn into a monitoring program of any kind. And so, and just to kind of remind ourselves of what the late 1990s looked like, all the data was collected on cassette tapes. So that's what we've got. And so now we have these new things coming in, this disease coming in, and we have to kind of start over and think about what's happening with bats now. So over 15 years later, after this inventory work, we're back and looking at bats in Washington's national parks. But in addition to recording their sounds, which we're not doing on tapes anymore, we are also surveilling for disease and using some newer technologies to do some bat tagging and tracking, which I will talk about to understand their winter ecology. So I'm going to talk about each of these in turn. So our disease monitoring program, our acoustic monitoring program, and then this newer work on bat winter ecology. So for the disease surveillance, what we're trying to do is basically catch bats in the springtime. So starting right around now in April, mostly April and May, as they leave their winter, where they've been over winter, which we still have no idea where that is, and come back into summer roosts or spring and maternity colonies. And a lot of times, the ones that we're able to really track are ones that are in buildings or bat boxes. So what we do is we then go out and we can capture these bats from these bat boxes or these building exits. And we weigh and measure them and assess their health. We protect the bats, we protect ourselves, and then we are able to swab them with a sterile swab, their wings and their body, to see if we're picking up any evidence of that fungus. We can also examine the wings at that time for damage. So if the fungus had kind of infiltrated their systems over the winter, you could see that damage in the wings at times. And then we let them go. So once a year, we get to hang out with some of our bats in the park. In Olympic, we focus on 2 sites where we've found these kind of maternity or summer or transitory roosts. The first one is the Hoh Ranger Station. We're heading there tomorrow. They like to snuggle up in the attic of the Hoh Ranger Station, starting about now and lasting through the summer. And the other site is the Staircase Ranger Station, where if you can see this, they are all huddled around the stovepipe on the exterior of the building. These guys don't hang around all summer. They show up, they're around for a couple weeks. We either get them or we don't, and then they're gone, and who knows where they go. So that's what we do for our disease surveillance, and we do the same thing in our other parks. In Mount Rainier, North Cascades, San Juan are all places that we have known colonies that we can monitor in the springtime. And for now, the only place where we've seen PD or seen evidence of white nose syndrome is in Mount Rainier. But like I said, it's kind of on the edges of everywhere at this point. Another large part of our work has been acoustic monitoring. So acoustic monitoring is super cool because it is non-invasive. And as I've discussed, the bats, they're very cryptic, they're very small, they're active at night. And so this kind of gets around some of those challenges by allowing us to be able to monitor their activity patterns, because each bat species has a unique call frequency and call sort of shape. And I'll show you some sonograms in a little bit here. So it allows us to understand the species distribution across these different parks and their phenology, or their kind of seasonal timings of their use around these parks. So this helps us gather this baseline information on bat populations before white nose syndrome hits and as it spreads, so that we can say, okay, we now have some understanding of some patterns of bat activity now. If white nose syndrome just spreads, are we seeing an impact, at least at that activity level? So it complements our disease monitoring. The first thing that we did was an elevational study. This work was done in 2019 to 2020. We are still working through the data, but I'll kind of describe it for you. Our idea here was to really focus on the 3 big mountainous parks and capture environmental, seasonal, and elevational gradients. So what we did was we set up these transects that can run from low elevation up to high, and we set up these acoustic detectors that have, again, these specialized microphones on them. So you can see one here. It's kind of hard to tell, but there's a long pole running up that tree, and the microphone's on the very top, and then the recording device is in a box at the bottom. And we set these up on water targets, so ponds, backwaters of streams, or lakes, alpine lakes, and as well as in these forest gaps, which are areas that are kind of where bats might commute out from where they're snuggled up under the bark during the daytime to where they're going to go feed and drink at nighttime. So we set up these stations along these different transects, and then would monitor them for a few nights at a time across the season. So trying to get an idea of some of that seasonal patterning of use. So you can see the forest gap type of environment on the left. So we had either, these kind of spacious, beautiful trees with big gaps in them, like in the Royal Basin trail here below, and then Deer Park is shown above, off the Deer Park Road, kind of in one of those draws between the forest. And then here's a couple examples, of course, of our high elevation sites, which were usually a good excuse to hike to a beautiful lake with a bat detector on your back. In Olympic, we also we wanted to check out a couple other sites we thought might be really important or neat to look at for bats. So we spent some time on the Elwha restoration area above the former Glines Canyon Dam in the Mills Reservoir, former reservoir. And then we also set up some detectors out on the coast, kind of from Lake Ozette out and along that triangle trail out there. So across all the parks, every single one of these dots is one of the locations that we set up detectors in one or both of the years. And then zooming into the peninsula in Olympic National Park, you can see our different transects, and I've kind of circled them here so you can imagine that we're just trying to cover all the diversity of the park, from like the wet to the dry, and then from low to high. So we've got, Royal Basin, Deer Park, and Hurricane Ridge, and then the Solduck and the Hoh and the Quinault and Staircase, as well as those kind of other sites I mentioned. So what happens then? So we've got our acoustic detector. with that specialized microphone, and all the data is being recorded on these cards that are in the detector. We pull out the cards, we download the data, and then we use a program called Sonobat that does two important things. The first thing is that it scrubs out all the rain. There is so much rain. And so we might get this is summer, but on a winter's day, we could get, hundreds of calls, but they're just rain files, and then maybe one bat. So that's the first important job, is getting all the noise files out. And then the second job is the program, actually, they have kind of set it up where you can use it in different, for the species you'd expect to see in different states. And so we have the Western Washington, and it will go through those call files and assign it to species or to a species group. 'Cause sometimes the call isn't good enough quality to be able to assign it to a species. So I'm going to show you a couple examples of that. So this is the California myotis, which is a kind of a West Coast-ish species. And it has this high frequency call and that we can't hear, but we can kind of see the shape of it. And you can compare that to something, and I picked some with really big contrast, like the hoary bat, which is a bigger bat species. It's one of our migratory bats and one of the ones that's impacted by wind energy. And it's got a much lower frequency call. And so you can see how those would be assigned differently. A lot of times it gets a little bit sketchier when you have a lot of myotis species have a lot of similarities. And so then our dear friends at Oregon State University and the Northwestern Bat Hub have been instrumental in helping us kind of confirm that the machine is seeing what we see. So we have experts down there that can look at some of these calls and do what we call manual vetting to confirm that species we're seeing are actually present. Okay, so we have 10 bat species here in Western Washington. We picked up all 10 species in Olympic at our sites, and we picked up 9 out of the 10 in the Cascades. The one difference was the Townsend's big-eared bat. This guy has a really low frequency call, and it's pretty quiet. And I think also where we had our detectors, it was just harder to pick up. Like, we're in the trees and it might be flying over the trees. But it's definitely found across Western Washington. So that's how many species we have here. We have 6 myotis species, and then we have silver-haired bats, hoary bats, big brown bats, and the Townsend's big-eared bat. I went back and forth. I did put a few graphs in here, so I'm going to spend a little bit of time walking you through those to just kind of show you a little bit of the variation between different species and different areas of the park. So I'm going to start, and I hope I'm realizing this is going to be a little bit too small for the back, but I'm going to start with the California myotis, which we talked about earlier. It is found year-round. It's active year-round and stays here year-round on the peninsula. And what I've done here for these graphs is I'm showing the number of bats per site night. So just kind of an index of activity, number of bat calls. And then on the bottom, we have our different study areas. So Deer Park, the Elwha, the Hoh, the Hurricane. And I don't want you to focus too much on the details. You're welcome to ask me questions later, other than to say that, in different parts of the park, we're seeing lots of variation in the activity of this bat species, with a lot more activity out in Ozette and out in the coast and over by Staircase than we're seeing in the Soldock or the Elwha, for example. We compare that exact same graph, index of activity, number of bat calls per site night is the numbers, and then these same sites in the same order. So you can see for the little brown bat that we just see a lot more activity in our wettest site in the Quinault, but much less in some of the places where the California myotis was really common, like Ozette and Staircase. I don't have an explanation for this right now. This is, again, our ability to kind of understand the baseline distribution and use of these different habitats. The little brown bat is one of the bats that have been most concerned about because it has declined so much in the east due to white nose. So it is quite vulnerable. Whereas the California myotis, I believe, is still, we've not ever detected white nose in California myotis. Two more species, just because it's fun. The hoary bat is one of the species that we saw the most. There's still really low numbers. So these numbers for those in the back go from 0 to 70, and this one goes from 0 to 7. So it's like a 1/10 of the numbers, but it's still, what you can see is that we're seeing them the most in the Elwha restoration area. And so that's kind of neat. So that riparian, that growing riparian area seems to be a nice place for hoary bats to be. And that is exactly the same for the silver-haired bat. Our other, so the hoary bats are migratory, and the other two stay here year-round. These guys move. The silver-haireds can stick around, but they also really, really like that riparian zone. So again, this is the Elwha, and now this goes from 0 to 300. So lots of use of silver-haired bats in the Elwha. So kind of fun, just seeing those patterns. We're doing some modeling now to kind of understand what that's going to mean, like what might be driving those differences that we see at different elevations and different precipitation, and over the course of the seasons. The last thing I want to show you for this stuff is we also are looking at the species diversity, the number of species detected. Like I said, you know, they all exist in all of our parks. But here, I'm also like stuck here. I can't go any further. This just shows June, July, August, September, October, and the number of species detected going from 0 to 6. And the colors represent different elevational bands. So interestingly, at our low elevation, we don't see that many species. And this is on our forest gaps. But in the middle elevations is kind of where we see the highest species. And then we see that movement where we're not seeing that much in June, and then it really peaks in that July through September. And then by October, you know, those migratory species are moving away, and maybe some species are starting to become less active, and we're not picking them up as much every night. So that's another part of understanding that seasonal timing. So again, right now, we're actively working on doing some more like statistical analyses of these data and being able to really try to synthesize these patterns a little bit better. We also have an acoustic monitoring program that is year-round. So these are much less locations, but we keep the detectors up all year round. So this can help us identify hotspots of bat activity. And again, it kind of is tailored towards the same questions of understanding that time of activity over time and across species. And the thing to bring it back to the disease question is that what we're really not sure is what happens after all this summer monitoring that we do. So what's happening that fall to winter period, how active, which species are active over the winter, when are they showing up in the spring? So this helps us get a bigger picture of that pattern. So we have one set up on the Elwha. That's an interesting place to study and great to go to. So I'm just going to show you a couple sites, and you can just see that the sites, you know, they look a little bit different throughout the year. So this one I just have winter, spring, and fall. We pick a nice open location that kind of shoots over the river. We have a detector set up out at the Mora campground area. So this James Pond, which isn't really a pond anymore, but kind of a wet meadow habitat, is a place that we've monitored. And then we've got one at Lake Crescent. And so I'm going to pause here and just show you a couple patterns that we've been looking at Lake Crescent, place I'm sure you've all visited. And so this kind of shows us the bat detections per night over the course of the year. And the different colors here are the different years. So we started doing some work in 2019, but really 2020 through 2022. So this is all the species combined. So this is just all the bat calls we might have heard per night, summarized by month, and then these different colors are per year. So you can see a huge variation between years. Something happened in 2020. I can't remember what it was. But I don't know. I don't know if COVID had anything to do with bats just going crazy at Lake Crescent, but there's sure a lot of activity that year. And then you can also see a little bit of that seasonal pattern where things sort of ramp up. There's these little funny little pulses, all that summer activity, and then it kind of dies off towards the end of the year. One more look, exactly same bat detections per night, months of the year. And this is looking at 2 species summed across all the years. And the 2 species are the purple is that California myotis. So that's the one that we see some activity every single month of the year. It also really likes Lake Crescent. There's something going on there. And then the Yuma myotis. And so you can just see that the different species, again, this is a way to kind of see the different patterning of what they're doing. So you see that kind of springtime activity this summer, and then much lower activity in the winter. So what we hope to do with this, sorry, I've got one more slide, is the whole rainforest. We have a detector set over a pond there. This is taken from behind the detector, so you can kind of see it shooting out and how things look across the season. I'm not going to spend a ton of time on this other than to say that we're doing the same thing. We've got this is just all the years, 2020 to 2022. The pulses are the summers, and then it kind of dies off in the winter. And then all the different colored bars are all the different species. So we're getting all these just descriptive patterns of how these bats are using the area. It's kind of cool on its own. It's going to be super cool if we can combine it with the kinds of detectors that are set up all over our region. So we're just one little cog, in this big, much bigger region where we're trying to understand these bats. And so our hope is to be able to integrate the kinds of data. People are collecting this kinds of data all over the place now in the Pacific Northwest, and to be able to put that together to better understand timings of migrations and how that might relate to wind energy, how patterns might be changing with disease, or just kind of trying to better understand what bats are doing out there, because it's hard. So that's that. And the very last thing I'll spend a second on, and then I'll wrap it up, is our Winter Bat Ecology Project. This is a new project. We are using MODIS to track bats in the fall and winter. MODIS is the Latin word for movement, and it is an automated telemetry network. And what that means, and it uses like these stationary receivers to detect tagged animals all over the world. So this is an example of, they're all over the world. This is kind of zoomed into North America. Every single one of these is a detector, a stationary receiving. Wow, I just did a word salad there. It's a place, it's where the receivers are located. It was originally set up to really be used to track migratory bird migrations. And so you could put a little radio tag on a bird, and you could track the timing and patterns of migration across the continent or across the globe. And now it's being used more and more for other applications like bats and even insects. So we set up our own humble station on the Glen Canyon Dam remnant. And so it is overlooking the restoration area on the former Mills Reservoir. So these are the antenna, and this is kind of the area that it can detect animals in. And this is an example of the MODIS tag. So this is your average paperclip. So you can see how tiny that thing is. And so what we can do is we can capture bats, and we can put these little tags on. They last for a few weeks, and understand how bats are moving around in the area. For our application, we're actually not trying to track migrations across the continent. We're trying to see how active bats are in the fall to winter period. So these again are our non-migratory bats, our little tiny myotis. And we're trying to understand how they're moving through the system in that period where they would start, where we just don't know much about their ecology. And so for this, we use mist nets that we just did, and this is fresh right in the middle of this project. And so it just got started last fall. And we'll trap again this fall, but we put mist nets up, these really fine nets that bats can fly into, or birds. And then we keep them in these individual little receiving bags. And then here you can see that little detector going on the back of that bat, and it will just shed it over time. So it's not like it's saddled with this tag forever. And what it does is it allows us to track so every time the bat's in the receiver, the bat's in the distance of the receiver, you can see it show up. So this is not something you need to focus on, but each color is a different bat. And you can we've tagged 10 bats last year, and then this is time, and then the hours of the day. So this grayer block is the nighttime, so we can see when they're popping in, popping out of the range of that receiver. And it's helping us early days, just kind of understand how they're using that space in that transition period. So this is in collaboration with USGS colleagues and parks in California as well. So we're looking at this across multiple parks, and we're going to install a couple more towers and try to understand what's happening to bats on all those times of the year we can't see them. So I'm going to wrap it up. Ultimately, we're trying to just better understand bat use on the peninsula and beyond. We want to understand and predict where these hotspots of bat activity are and measure change when disease becomes more widespread. So a lot of what we've done until now, until this MODIS project, has just been, how are bats doing in this kind of mostly disease-free landscape that we have? And how can we use that to understand how things might change moving forward? And to understand the ecology of bats in the Pacific Northwest, which are we just don't know that much. It's just kind of amazing. And of course, to share that information with all of you. So happy to answer any questions you might have.
Thank you so much. So if you have a question, if you're watching at home and you have a question, please leave yourself muted, but you can put your question in the chat and I will read it out as we have time. Or if you're here in person, you can raise your hand and Audra will bring you the microphone. And while you're thinking of your question, I will start with one. Why or what are the plans for doing bat research at Mount St. Helens?
Oh, well, I have no idea. So, yeah. Oh, of course. Yeah, asking about whether or not if there's any plans or known work happening at Mount St. Helens. And my answer was, I don't know, personally. Yeah.
Hi, do you know how the white nose syndrome hopped across the country like that? Seemed like it was in the east and then all of a sudden it was, in Washington, and the whole middle of the country still there wasn't any.
Yeah, well, unfortunately, sometimes diseases, I don't think there's no answer like that's been, but probably people. So some of these emerging diseases, you've got people who like to go caving. It's just contact and accidental transmission of something like that. And it just, it caused a jump. So, yeah.
If you capture one with the white nose, is there anything you can do with it before you release it? If you tag it and you know it has that, is there anything that can be done for the bats to stop this spread at all?
Yeah. I'll put a little mask on it, yeah. So there isn't right now. I mean, there has been some work that is really an active area of research development right now, to either prevent mostly it's to prevent them from getting infected. It's just hard to think about how you would apply something like a vaccine or something of that nature at any kind of widespread level in wild populations. But there are, again, it's just kind of like an active area of work. So, but yeah, I mean, once they have it, they have it. It really is kind of integrated into their bodies.
Couple of questions from online. I'll share two and then we'll go back to the audience. One, are you monitoring bats for rabies? And if so, how often do you detect it? And two, where do migratory bats go when they migrate?
Good questions. So for the rabies question, we don't monitor bats for rabies. It's not very frequently found here. The two incidences I have heard about ever in the park was one bat, Lake Ozette, 2008. And then I think the previous one was in 1974. So this is not something compared to some parts of the country that has been a huge problem. We certainly, as you saw, protect ourselves and protect the bats. You know, the masks came in around COVID because we were afraid that we could give COVID to bats, you know, so we do protect ourselves and we're all vaccinated for rabies. Don't touch bats, like don't do it. So, you know, if you have a bat that you have a concern about, don't just pick it up and bring it to somebody. But it hasn't been a major issue and none of us has encountered a rabid bat during the course of this study. The other question was about migration. So the hoary bats and silver-haired, they go south. They go to California and Mexico, and it is an area again of like kind of active research because we haven't really, these technologies that we're using now are all relatively new, and I think the wind farm question has really increased the need and urgency for understanding those migration patterns in a way that maybe we hadn't studied previously. There's also been some work just looking at some interesting patterns of like how bats might just move from like the mountains to the sea, you know, so there might be some more dynamic things going on where bats are actually staying more local than we think in some cases. So yeah, so still a lot to learn. But definitely MODIS has been used to document movements of bats from California to Washington.
There's a plethora of information on wind farms impacting birds. And is there any attempts to try to digest that data to see if it's an impact on the bats?
Mm-hmm. Yeah, I mean, it's a similar threat, right? There are different things about how bats navigate through space at night compared to birds flying during the daytime. But there's work being done. And I think the two systems are informing each other. I certainly wouldn't consider myself an expert on that particular area of work and research, but yeah,
There's another piece to that.
Mm-hmm.
There's another piece to that puzzle, and that is that we're just now finding out that lights at night are affecting the navigational capabilities of insects.
Mm-hmm.
You know, because somehow or other they give the orientation, and do the lights at night, you know, we're living in a paranoid world. Everybody's got lights on all over the place, you know, and is there any evidence that the light pollution that we're inflicting on our environment is impacting the bats?
Ooh, I have not heard about that personally, which is not to say that it's not an issue. Yeah, I was going to say that with the wind farms, there is you know, some of the things that people are looking at, and I'm sure it's the same for birds, is, you know, the timing of migration. And so can we adjust when they're operating if we know there's like the peak of migration of animals coming through? There's also work being done looking at how fast the blades spin and, you know, so there's like a lot of pieces going into that, and it's for both birds and bats. Yeah. Yep. Mhm.
So I have a question about how do you attract a bat? I lived on the East Coast, so I was real familiar with the white nose syndrome because it was starting up then. And I've never seen bats here. You see them all the time, but I've never seen them like in my backyard or coming out of my garage, or I'd like to have a few. So is it,
You probably do.
think I might?
Yeah.
Okay.
Yeah, I don't know if there's, you know, it's funny because we were talking about this before the talk started, about attracting bats. I mean, they, you know, ideally they would have the natural habitat and they wouldn't need to use our buildings and our eaves and our, you know, different parts of our human habitat, but those can also be great places for them if people are happy to have them there. But you know, they're going to pick the spots that they're interested in being in. Yeah. Yeah. I mean, I would say, you know, plant a pollinator friendly garden and attract as many insects as you can, because we need more insects too. Yeah.
One more question from the online audience. Ian says that early season spike in detections at Lake Crescent is interesting. Do you think it is related to some availability of a particular prey early in the spring? And then a separate question, how did removing the Elwha Dam affect bat behavior? Another twofer.
Yeah. So the Lake Crescent question is interesting. So I have to preface that when we do acoustic monitoring, we could have one bat and we could pick up that bat flying in front of the detector all night. And there have been instances where people have actually been like, oh, there's not that many bats out here. There's just a couple bats flying in front of this detector over and over, versus, which is also very likely, that there's all kinds of bats coming in, drinking and leaving, and drinking and leaving. So we can't really tell. One thought is that there could be a maternity colony near there. And so you're starting to get kind of a larger aggregation showing up right at that around this time of the year, that kind of March to April period. But it's hard to know. It's just also hard to know exactly what we're capturing there. And I think that's one of the challenges of acoustic data, that we, you know, are trying to do our best to kind of parse out. So regarding the Elwha, we don't know because nobody was looking at bats before the dams came out. What we have with those dewatered lake beds is this really active riparian, like actively revegetating riparian zone. And so it is a different kind of environment. So you can imagine that it's a pretty sweet spot right now, because the bats could be in that beautiful mature forest all around the dewatered lake bed and spending their daytime behind little crevices of bark or up in the trees. And then they come out to this like rich tapestry of insects and riparian vegetation and water at night. But when the reservoir was there would have still been a bunch of water, but different vegetation, which is probably, you know, very different for the insect community. So.
Let me speak loud.
Use the
Do they hibernate anyway?
Do they hibernate? Yes. So they do. They go into this kind of hibernation state or torpor in the winter time. Some bats are more active in the winter than others. So like I said, the species like that California myotis, we do pick it up all winter. So it seems like it has maybe shorter times before it gets back up. and needs to drink or just starts moving around again. Whereas other species go into more prolonged time periods where they're hibernating. We really don't know much about what happens with bats in the winter here. It is like a wonderful ecological mystery. In the East Coast, you have these huge caves and you can have your thousands of bats in there. And we don't really have those environments in the same way here. Our best understanding right now comes from just a handful of studies, and it's that bats might be alone in groups of 2 or 3, maybe up to like 10, and they're in little, root holes and behind the bark and maybe in rock crevices. So just like a totally different dynamic. So
We're going to take 2 more. Last 2.
I was wondering, do bats have, like danger calls, like other birds sound an alarm, if there's a hawk above them or something? And if they have those kinds of tones or vibrations or whatever, I wonder if the spinning blades of the machines, the wind machines, whether you could recreate that warning call, and the bats would go higher or go left or go right, and maybe stay away from the danger areas.
Yeah, that's an interesting idea. I don't think, as far as I know, bats do not have warning calls. As far as I understand, the spinning of the blades does super duper mess up with their echolocation. And so they're spinning too fast for them to like see where they are. And so that's kind of like part of the issue, is that, they might hit a blade and then they decide to go left, and then that's where the blade's going, something like that. So that's, yeah. So.
Why not? Hi, are there things that we do in our day-to-day lives that impact the bat community that we might not be aware of? Like using pesticides or plastics, or I don't know, just anything we should be aware of?
I think the best thing that we can all do from day to day is to try to promote insect life. They are another really understudied group that has shown dramatic declines. And all our bats around here eat insects. And so, yeah, having those, garden spaces and those natural spaces, and not applying pesticides, and, letting the mosquitoes feast upon your flesh while you're out camping. Let the bats take care of them.
Somebody online did chime in and say they've heard that light pollution causes energetic costs as bats toggle between visual and echolocation when foraging. So thank you for including that. More bugs, less light. Sounds good. Thank you. What a great talk. Let's have a round of applause. Thank you.